For decades, Beringia was taught as a frozen shortcut, a simple land bridge crossed once by a single wave of people on their way into the Americas. But in 2013, two infants buried beneath a hearth in Alaska proved that story was wrong. Their DNA didn't just challenge migration theories. It revealed a lost population that lived, evolved, and vanished at the edge of the Ice Age. At the bottom of a circular hearth in what had once been an early Native American dwelling, they found the skeletons of two infants lying side by side. One infant, later designated USR-1, had died between 6 and 12 weeks after birth. The other, USR-2, was a late-term fetus at approximately 30 weeks gestation, who had apparently been stillborn or died shortly after premature birth. Both were covered with an undisturbed layer of red ochre, a mineral pigment associated with burial ritual and ceremony across cultures. Then scientists sequenced their DNA, and what they discovered proved something remarkable. These infants belonged to two completely different genetic lineages, and that revelation would rewrite our entire understanding of how the Americas were populated. Radiocarbon dating and stratigraphic analysis revealed the age of this burial, approximately 11,500 years old, making these the earliest human remains ever found in the northern reaches of North America. The infants were not alone in their grave. 4 decorated antler rods, 2 lithic dart points, and additional stone tools accompanied them. The antler rods and dart points likely formed part of a weapon system or hunting implements used to bring down large game that sustained these early peoples. The burial pit lay directly beneath another remarkable feature: the cremated remains of a 3-year-old child, found within the central hearth and dating to the same period, discovered in 2010. The two burial events were contemporaneous, dating to the same brief period, yet their treatments diverged sharply. The 3-year-old was cremated in an elaborate ceremony, while the infants were carefully buried with grave goods intact. Together, the three represent to date the only confirmed human remains from that period found in the area that was once Beringia. But the true revelation of the Upward Sun River discovery would come not from the bones themselves, nor from the artifacts buried with them, but from the genetic information held within those ancient remains, their DNA. A team from the University of Utah undertook the work of extracting and analyzing genetic material from the infant's bones. They focused initially on mitochondrial DNA, the genetic material contained within mitochondria, which passes only from mother to child without recombination, creating clear maternal lineages that can be traced back through time. Despite the age of the remains, the team succeeded in sequencing the complete mitochondrial genomes of both infants. The cremated remains of the 3-year-old child could not be genetically analyzed as the intense heat of cremation had destroyed any DNA. When they analyzed the sequences, they made a crucial discovery. The two infants buried together belonged to 2 different mitochondrial DNA haplogroups. Haplogroups are major branches of the human genetic family tree, defined by shared mutations that indicate descent from a common maternal ancestor. USR-1, who had died between 6 and 12 weeks after birth, carried the C1b lineage, a genetic signature found today among indigenous peoples from the Great Lakes and northeastern woodlands of North America, such as Ojibwe and Abenaki, to tribal communities across the Amazon basin in Brazil, Peru, and Colombia. USR-2, the stillborn, carried the B2 lineage, another distinctly Native American genetic marker that appears across indigenous populations from the Arctic of Alaska and northern Canada through Mexico and Central America and into South America, all the way to Patagonia in southern Chile. To understand the significance, we must examine the larger puzzle. Of Native American origins, indigenous peoples of the Americas belong to 5 major mitochondrial DNA haplogroups, designated A, B, C, D, and X. The presence of 5 haplogroups raised an obvious question. Did this diversity result from multiple waves of migration from Asia at different times, with each wave bringing a different haplogroup? Or could there be another explanation? Some researchers had proposed an alternative hypothesis, one that would come to be known as the Beringian Standstill Model. It posits that a single major wave of people entered Beringia from Asia and remained there for thousands of years. And during this prolonged isolation, cut off by distance, climate, massive ice sheets, and other factors, the Beringian population differentiated genetically. New mutations arose and spread. Genetic drift created variation. The 5 haplogroups didn't represent 5 separate migrations, but genetic diversity that evolved within Beringia itself. The Upward Sun River infants provided direct evidence for this model. Both C1b and B2 are common in Native American populations, but non-existent in Asia, strongly suggesting these genetic markers arose after the ancestral population had entered Beringia. Using molecular clock techniques, a method that estimates the age of genetic lineages by measuring how quickly mutations accumulate over time, the team estimated C1b arose approximately 12,800 years ago, and B2 around 12,000 years ago, after entry into Beringia but before expansion into the Americas. Now, an international team has sequenced USR1's complete nuclear genome. The results were even more remarkable. USR1 was genetically different from all other Native Americans, both ancient and modern. Her DNA shows she belonged to a unique ancient Beringian population, one that lived at the same time as the ancestors of today's indigenous peoples, but followed its own separate genetic path. Through sophisticated demographic modeling, researchers pieced together a detailed timeline. The ancestral Native American population had already formed as a distinct group, shaped by long periods of separation and mixing with other ancient peoples. With this genetic foundation in place, they moved into Beringia, where ice sheets blocked passage south and harsh conditions limited other movement. Around 21,000 to 20,000 years ago, the Beringian population split into 2 groups. One became the Ancient Beringians, represented by USR1. The Ancient Beringians remained in Alaska for thousands of years, adapting to harsh conditions. Meanwhile, the other branch eventually moved south. But what happened to the Ancient Beringians? Genetic evidence suggests that after 11,500 years ago, people carrying northern Native American ancestry moved northward back into Alaska. This back migration may have replaced or absorbed the Ancient Beringian population, whose distinct genetic signature disappeared from the record, even as elements of their material culture persisted. Yet, even as these genetic revelations support the Beringian Standstill Model, new archaeological evidence complicates how people entered the Americas. The ice-free corridor, once thought the main route south, did not become biologically viable until about 13,800 years ago, after humans had already reached far southern sites. So, it was closed during the initial spread. This strengthens the coastal migration or kelp highway hypothesis, in which maritime peoples traveled down the Pacific coast using rich marine resources. However, much of this coastline is now submerged, making study extraordinarily challenging. The schoolyard theory of Beringia, as a simple land bridge, has indeed been fundamentally challenged. Beringia was not merely a highway, but rather a homeland in which Native American genetic identity was forged. The Upward Sun River infants sit at the center of these debates. They are the most important human fossils from Beringia, a region vital to human prehistory, yet yielding very few ancient skeletons because most of it is submerged, glaciated, or chemically hostile to preservation. These infants are virtually the only remains from this period with high-quality genetic data. As research continues, new discoveries and improved analytical techniques will refine this picture, ensuring that our understanding of the first peopling of the Americas will continue to evolve.